This method can help answer key questions in the microbiology field about how often plasmid DNA can be spread among different bacteria. The main advantage of this technique is that by reducing the background, we can detect small differences in the conjugation frequency with a high resolution. Demonstrating the procedure will be Kosuke Yanagiya, a graduate student from my laboratory.
To calculate the conjugation frequency by most probable number, filter mate 1 milliliter from an overnight donor culture with 1 milliliter from an overnight recipient culture for 45 minutes at 30 degrees Celsius, as just demonstrated. While the co-culture is incubating, serially dilute the original donor and recipient cultures for plating in triplicate onto donor luria broth, or LB, plus kanamycin, or recipient LB plus gentamicin plates for a 2-day culture at 30 degrees Celsius. At the end of the incubation, resuspend the culture on the filter in sterile LB containing kanamycin and gentamicin for serial dilution in a 96-well cell culture plate in quadruplicate. After 2 days at 30 degrees Celsius, manually count the colony-forming units, or CFUs, on the donor and recipient agar plates, and the number of wells in which the transconjugants grow by light microscopy. To calculate the most probable number and its deviation, open the MPN calculation program. Enter the name and date of the experiment, the number of the test series, and the maximum number of dilutions in row 7 of the program sheet of the spreadsheet file. In the automatically produced input data tables, enter the formula in the dilution